Here we have a muscle. Then we pulled out the fascicle. Inside the fascicle is a muscle fiber, and inside the muscle fiber is the myofibril. Action potential arrives at synaptic knob. Calcium channels in membrane open, allowing calcium to move into the cell. Increased calcium causes synaptic vesicles filled with acetylcholine to fuse with the membrane. Acetylcholine is released from the synaptic knob by exocytosis into the synaptic cleft. Acetylcholine is picked up by acetylcholine receptors on the motor end plate of skeletal muscle. Sodium and potassium channels open, allowing sodium ions to enter the muscle fiber and potassium to enter the synapse. Acetylcholine esterase is in the synapse and breaks down acetylcholine into acetic acid and choline to end the cycle. An increase in positive ions inside the muscle cell initiates an action potential on the sarcolemma. Action potential travels down the sarcolemma into the T-tubules and stimulates the release of calcium from the terminal cisternae of the sarcoplasmic reticulum, where it has been sequestered by calsequestrian into the sarcoplasm of the muscle cell. Calcium floods over the thin filaments and binds to troponin, pulling tropomyosin off the active site on the actin. High-energy myosin heads leap to bind onto the exposed active sites on the thin filaments, forming a cross bridge. The myosin head pivots to the low-energy state, releasing ADP and moving the thin filaments over the thick filaments. Mitochondria produces ATP. ATP binds to the myosin head, causing the head to break free from the active site on the actin. The myosin head becomes energized as ATP is converted to ADP. Calcium is pumped back into the sarcoplasmic reticulum and attached to calsequestrian, allowing tropomyosin to once again cover the active site, resulting in relaxation of the muscle. Within myofibrils are smaller structures called filaments. There are thin filaments and thick filaments. The thin and thick filaments are arranged in compartments called sarcomeres. Sarcomeres are the basic functional units of a myofibril that produce muscle contraction, relaxation, and stretching. Z-discs separate one sarcomere from the next. The thick and thin filaments overlap one another to a greater or lesser extent depending on whether the muscle is contracted, relaxed, or stretched. The darker middle part of the sarcomere is the A-band, the zone of overlap of thin and thick filaments. The I-band is a lighter, less dense area that contains the rest of the thin filaments and no thick filaments. The H-zone is in the center of each A-band and contains thick but not thin filaments. The M-line is located in the middle of the sarcomere and supports proteins that hold the thick filaments together.